Good afternoon. Welcome to GENEWIZ Week and today's webinar, Genome-Wide Synthetic Biology, From Pathways to Genomes. I'm Damian Urena, Business Marketing Leader for Next Generation Sequencing at GENEWIZ. I am pleased to introduce our featured speaker for today's session, Dr. Marc Güell from the Wyss Institute at Harvard University. Dr. Güell earned his PhD in biomedicine from the Center for Genomics Regulation Research Institution in Barcelona. Recently, as European Molecular Biology Organization and Human Frontier Science Program Fellow in the lab of George Church, he focused on genome recoding and in CRISPR-based genome engineering. Currently, as a Wyss Institute Technology Development Fellow, he focuses on synthetic systems to generate human-compatible tissues and to translate DNA diversity to small molecule diversity. Please join me in welcoming Dr. Güell. Please note, if you have any questions, kindly use the chat room to submit them, and Dr. Güell will address them at the end of the session. Without further ado, Dr. Güell, welcome.
Excellent. Thank you very much for the introduction. So yes, basically today I'm going to present two different projects. But basically what they have in common is that we are altering properties, genome-wide properties of these two genomes. So basically the first project is humanizing pig genome for xenotransplantation, and the second project is I'll describe the synthesis of a radically recoded E. coli genome. Okay, so I'll start with the first project, basically the humanization of the pig genome. So maybe I'll start explaining a little bit the motivation behind this project. Basically, why do we want a humanized pig? So basically, two groups of motivations. The first one, and probably the main one, is basically that probably the biggest unmet medical need is probably the lack of tissues and organs for transplantation. There is a very big and growing list of patients waiting for tissues to basically to cure their diseases. So the pig has been proposed as an interesting animal because basically in terms of size and in terms of physiology, it's relatively close to the human. So basically, the pig could be a good source of tissues for cellular therapy, for example, beta isolates, neuronal cells. So it has been proposed as a potential source of vascularized organs, for example, kidney, liver, or others. In addition to that, we also have a second motivation. So basically, the pig is also a very interesting animal for research. So the pig has a closer proximity to human compared to mice, which is our traditional model in biomedicine. And it has been proven for many applications that it can provide very big advantage to build disease models, also to test drugs, and also to study embryology. However, in order to use the pig as a source for tissues for regenerative medicine, we need to face at least two main challenges. One of them is that there is a difference in evolution between pig and human. So basically, tissues from pig, when transplanted into human or primates, basically they trigger an important rejection. So one of the goals from this project is to increase the immune compatibility between pig to human. So there are a reasonably big list of incompatibilities. Basically, there are certain immune mechanisms that they are not fully compatible between primate and pig, that basically we are using genetic engineering to address those, basically to replace in the pig or to add in the pig human versions of certain immune and coagulation components to make it more compatible. And then also the pigs contain certain antigens that trigger a big immune response when transplanted into human. And we are also using gene engineering to eliminate these antigens. However, there is also a second block of concern, which is associated with the pathogen transmission. So, like all mammalian genomes, pig contain endogenous retroviruses, and actually the porcine endogenous retroviruses have been shown that they can infect cells. And basically, one concern that has arisen is that in order to create a safe pig tissue, it should have no active endogenous retroviruses so that they cannot jump from pig to human. So today I'm going to present in detail this part. Basically, I'm going to describe how we inactivated all porcine endogenous retrovirus into the genome. Basically, all the work of basically increasing the immune compatibility pig to human, this work is being packaged into GENEWIZ, a startup that has emerged from the Church Lab. And actually, you can find more information in this website. So basically, what are the PERVs, the porcine endogenous retroviruses? Basically, they are endogenous retroviruses, so they reside in the genome. These type of viruses, these specific viruses, they are not infectious in the host, but actually they may become infectious in other species. So that's the reason why there was this concern about the potential of infecting human cells if these big tissues used for transplantation. And then since these viruses reside in the genome, no matter how clean we breed our animals, they will always be there because they are transmitted vertically from parents to offspring. Here in this phylogeny, I'm showing the different endogenous, the different retroviral elements in the pig genome, and here I'm highlighting what are the porcine endogenous retroviral elements, the ones that have been shown to infect human cells, and that I'm going to describe how we eliminate them, inactivate them. So, as I said before, this has been a concern in the xenotransplant community, and actually this was the reason to stop different medical programs due to the concern of these viruses to jump from pig to human. Basically, these viruses are composed by 3 elements flanked by LTRs, so 3 different genes, GAG, and END. And they are normally present in multiple copies into the pig genome. For example, in the cell line that I'm going to discuss today, we have about 62 copies as measured by digital PCR in this plot. So how did we basically plan the elimination? So basically, this is the scheme describing the genome of the endogenous retrovirus. Basically, it contains these 3 genes and is flanked by some LTRs. And actually, what we did is we designed some very specific gRNAs. I'm going to describe in detail how we designed these gRNAs that target one of these genes. And then we package these gRNAs into a vector together with Cas9. And this vector is a piggyback transferase vector that gets inserted into the genome. So basically, how did we design these gRNAs? So basically, I just will remind that these PERVs are in multiple copies, so it presents some additional challenges since we are editing a repetitive region, and we will cause a much higher genome damage. So it's very important to design gRNAs that are comprehensive, that they target all the copies that we want, but that so that they are comprehensive, but also they are specific, that they do not target to minimize minimal damage in other parts of the genome. So what we did is we did a multiple genome alignment of all the endogenous retroviral elements in the pig genome. Then we identified the functional site, so the catalytic peak site of the pol gene, which is an essential part of the endogenous virus. And then basically we developed an algorithm to design specific and comprehensive gRNAs to inactivate all the copies, but to minimize basically the off-target activity. So this is graphically how it looks like, our design. So these are our gRNAs. The gRNAs that we designed. So basically, they target this very conserved area. Also, I would like to take the opportunity to highlight that these viruses have some variation, so they have different haplotypes. I'm bringing this up because we will use it later to analyze the gene editing results. So basically, this is the phylogeny of these different haplotypes. So here in yellow, in these yellow stars, I'm showing basically the abundance of each of the haplotypes. You see that there are two haplotypes that are quite abundant, and then we have other haplotypes that are less abundant. So this is how we did the experiment. So basically, in the beginning, so we used the construct that I described before, this piggyback construct containing 2 gRNAs and Cas9. So we transfected this construct into the cell. We selected for it because it also carries a pyromycin selection to select the cells that integrated this part into the genome. And then we started doxycycline induction to induce Cas9 expression. So, and in different days, we collected cells and we measured how much targeting we had achieved in these cells. As you can see, we see a maximum level of targeting in day 17, which is about 34%. We took the cells, this population of cells, and then we sorted using FACS single cell sorting. And here is where it started our first surprise. Actually, we saw a bimodal distribution of cells. So we saw that the majority of cells didn't have editing, so they were almost completely active. But then we have a few clones that actually were completely inactivated or mostly inactivated. Here is the same results, so more details on the same results. Here I'm just bringing up again the phylogeny. So just to remind that we have these different haplotypes of viruses, and these are represented in different purple shades. So each of the different purple shades represents different haplotype. And in red, I'm showing the deletions. So basically, in the x-axis is the genome of the virus, and basically when we see red, this means that these are reads representing inactivated viruses. In the population, we have about 36% inactivated, and then when we sort into single clones, we detect clones and they have 100% activation. You see that the red elements, they go all the way from in all the element sequence. And we also have clones that have almost no inactivation. We were quite surprised to see this bimodal distribution, and we basically explored which could be the reasons to have this phenomenon of seeing a few clones highly edited and a big number of clones not edited at all. So basically, one of the potential explanations is that there is a significant gene conversion happening. Basically, that the first edited alleles, that basically they are used as a template to repair the next consecutive alleles. And actually, this is supported with some data. I'll show you later. Then also, we wondered whether the highly modified clones, they had some specific genomic or epigenomic modifications. We did some RNA-seq and some whole genome sequencing. We did not observe dramatic changes or obvious changes that could explain the higher editing efficiency. And then another factor, which is simple, but actually it's definitely an important one. So basically, that the clones that express more Cas9, normally they are associated with high editing. The fact that different clones express different Cas9, it's explained by, we are using, I just want to remind that we are using the piggyback transposase to deliver the Cas9 and the gRNAs, and this is a random integration mechanism. So depending on which area of the genome the Cas9 gets inserted, it may give different expression levels. So here I'm just showing some more details on these different hypotheses. So the fact of gene conversion. We believe that we see some evidences of gene conversion, and the evidence is that, for example, we see the same indel. For example, this indel, which is a single base insertion, we see the same indel happening at different haplotypes. This means that very likely that could be that this was initiated in one of the lost sites and then propagated into other lost sites, supporting the gene conversion mechanism. Actually, we did some statistics on that. So basically, we analyzed basically the different indels present in one clone and compared with the different indels across clones. And actually, we see a high co-occurrence of indels inside the same clone, but we see very little overlap, very little co-occurrence between different clones, which is may explain that basically the different alleles in the same clone are correlated. So it would also support this gene conversion hypothesis. Then the other fact that I explained before, Cas9 expression. So all the highly edited clones express higher levels of Cas9 compared to the lowly edited clones. So this also seems an important factor. We built a model to study the mechanism of this editing process. So basically, we built a simple Markov model, so that each of the different states is represented by a certain level of editing. For example, one virus copy inactivated, 2 virus copy inactivated, and so on. So basically, the state 62 is a state that has 62 copies inactivated. The state 0 is one that has 0 copies inactivated. And basically, the different ways to, and the different changes that from a state to different transitions from a state to state that could happen are basically that a cut is relegated without mutation, so a Cas9 induced cut. So basically, then is a transition to the same state. Also, there could be a cut with mutations. It could be a transition between one state to the other. And also, we could also have a cut followed by template base repair. This would also basically jump from one state to the other. So basically, we simulated this model using different parameters of efficiency of template base repair and also non-homologous chain joining. And basically, and also we ran the simulation using different number of cuts, which the different number of cuts emulate the levels of Cas9. So the more Cas9, the more cuts. So and actually we see that basically when the major factor is the number of cuts. So basically having more Cas9 seems to be the very important factor to get a high efficiency in the editing process. Here in green means 62 mutated perps, and pink means basically 0 mutated perps. So then after demonstrating that we could genetically inactivate all these retroviral elements, what we did is we test functionally whether the cells that had all the endogenous retroviruses inactivated, whether they were capable to infect human cells. So we co-culture pig cells with human cells, and then we check whether the pig cells had infected, so the perps from the pig cells had infected the human cells. For example, here I'm showing the data for clone 20. Clone 20 had all the retroviruses inactivated, and actually we could validate that this clone couldn't infect the human cells. However, clone 40, that was a clone that didn't have the perps inactivated, basically it could infect the human cells. Yeah. And then another concern of this project is that since we are modifying so many loci of the genome, we're concerned that we may be getting a little bit of off target. So what we did is we studied the regions that are 1 or 2 mutations away from the GENEWIZ that we designed to target the perps. Actually, here I'm just showing plots of whole genome sequencing data demonstrating that we couldn't detect indels in these positions. Actually, the only place where we could detect indels was in the direct site. Here I show it in red. So we did not see evidence of off-target in this experiment. Although I should mention that the pig genome is not, basically it doesn't have a very high coverage, so we may be missing something. But yeah, basically here we use our best effort. In summary, basically we demonstrated that we could inactivate all 62 copies of reinforcing endogenous retroviruses from the pig genome, and we could show that the edited clones had no infection capacity. We think that this shows us a method to create a safe cell line for using in transplantation, but also we think that this method could be exported to other repetitive regions of medical interest, for example, line ones or other repetitive elements. So this is what I wanted to share you about the pig project. So I'm going to switch gears to the second project, which is basically still involves modifying genome-wide properties, but also in this case from a bacterial genome. I'm going to describe the synthesis of a radically recoded E. coli genome. So to start, maybe I'll start explaining the motivations to synthesize a radically recoded E. coli genome. And also I would like to also explain what I mean by recoded. I mean, so basically recoding in this project means basically changing the genetic code, so eliminating codons from the wild type genome. Basically, if we eliminate codons, if we recode the genome, we have the opportunity to introduce non-synthetic amino acids. This basically has advantage that we could generate an upgrade of life chemical space. We could introduce new amino acids with new chemistries, new labelings, new structural motifs. Probably even more important than that, if we generate a bacterial species that has a different genetic code, basically we can generate what is a biocontained microorganism. Now, every time more, we use and we propose to use bacteria for medical therapy, for environmental remediation. And it's very important that what we release into the environment or into our body, that we control the propagation. So in the case of having a recoded genome, this DNA cannot be shared with other species, and also it allows us to introduce synthetic alternatives. For example, this was demonstrated by my colleague Dan Mandel in the Church Lab. Basically, he demonstrated that he could redesign an essential genes of E. coli to be dependent on non-natural amino acids, and then basically he could control the propagation of this bacteria by the supply of the non-natural amino acid. And then the third motivation is viral resistance. So basically, if we have a bacteria that has a different genetic code, it will not be able to be infected to viruses. It will be immune to viruses. The principle of this concept was demonstrated also by my colleague Marc Güell in 2014. That basically, he showed that with a strain that had one codon recoded, he could demonstrate that is more resistant to this type of infection. In this project, what we are eliminating 7 codons. We are eliminating 2 arginine codons, 2 leucine codons, 2 serine codons, plus one stop codon. This implies a big number of modifications in the genome, as I'll show later. So this is not the first genome refactoring project. So there was from the venture team the synthesis of a mycoplasma genome, which was very little recoding, about 1 megabase of synthesis. Also, a more significant percentage of recoding was achieved by UND's lab in the refactoring of the phage T7, where they recoded about 3% of the genome. And then also from the Church Lab, basically, a few years ago, it was generated in a strain of E. coli that had one stop codon eliminated, which it's a relatively low recoding of a quite large genome. So if we compare the current project, 7 eliminations of codons, so we are refactoring about 3.8% of a 4 megabases genome, and compared to the other recoding projects. So this is the representation of how many changes we need to introduce into the E. coli genome to achieve the 7 codons elimination. I'm showing the y-axis the number of changes, and the x-axis the position in the full E. coli genome. Is more than 64 base changes. With current technologies, it would be very difficult to get these changes by gene editing, so we had to synthesize the genome from scratch. So basically, this is our workflow. So first, we designed the genome on the computer. Basically, we eliminated the 7 codons. Then we synthesized the full genome in small pieces, and we assembled these small pieces into bigger fragments that more or less represent 1% of the E. coli genome. Then we transform these pieces into wild-type E. coli and then generate what we call a duplication. We have the synthetic part and also the wild-type part, and then in a subsequent step, we're eliminating this part. So basically, we could demonstrate the design principles that we applied. They could create viable strains, and basically we have a manuscript under revision that is describing basically how we could basically establish this pipeline and generate this radically recoded genome. So with that, I would like to close and thank my mentor and supervisor, George Church, for having generated an extraordinary environment to do science, and then also the GENEWIZ team that I have that was involved in the humanization of the pig genome, and also the RC57 group that was involved into the synthesis of this radically recoded genome. I'd also like to thank all the other lab members, the funding sources, and thanks to you for your attention.
Thank you, Dr. Güell, for your presentation. We will now open it up for questions. Again, please enter your questions into the chat room, and we will address as many questions as possible. Okay, we have a question coming in here. Marc, how did you decide how many and which codons to eliminate from the E. coli genome?
Hi, do you hear me?
Yes.
Hello. Okay, perfect. So actually, that's a very good question. So basically, there were multiple factors involved into this decision. So first, we wanted to modify enough codons so that the biocontainment of the strain and the viral immunity would be big enough, and 7 we believe that provides this property. And then with 7, we first, of course, try to minimize the number of changes, and we also, another important criteria was to basically use codons that we already had experience with. So, for example, the stop codon is a stop codon that we already eliminated in a previous strain in the lab, and also we have basically also lots of experience with arginine codons. So this was like the combined reasons of basically selecting those specific 7.
Okay, thank you. We have another one. This one is related to the pig genome project. Pig valves are already used for human heart surgery. Why are pig organs different from pig valves when it comes to human transplants?
Yeah, actually, the biggest difference between valves and, for example, other tissues that have been proposed for therapy, like beta islets or vascularized organs, is that the valves are decellularized, and this makes them much less immunogenic because of the absence of cells. That's why they have been used for a long time with much less challenges.
Okay, thank you. I think we have one more here related also to the pig genome project. What issues remain to be addressed for hospitals to offer pig organ transplants? And how long do you think it will take to resolve these issues?
I think the main 2 problems are for sure controlling this immune rejection, and for certain organs probably will be also very important to ensure good physiological compatibility, for example, with liver or and then the other big issue is to to make these organs safe, especially in terms of pathogen transmission. So both from the side of clean breeding, so providing animals that do not transfer exogenous pathogens, but also I think cell lines and animals that are free of endogenous retroviruses. I think they will add also another layer of safety. And then regarding the other part of the question, so how long? I think it's a bit difficult to say. Definitely, transplantation has had a very long history. Animal tissues have been engrafted into humans since the 17th century. First was more blood, the skin, but very often with not very good outcome, as you can guess. However, in the modern era, probably we are more seeing that first, these organs, especially the vascularized organs, they will need to be fully tested in primate models before going to the clinic. I have to say that for cellular therapies, it has been much more advanced. So there have been already clinical trials of beta isolates therapies for diabetes using basically beta isolates from pigs. But for the vascularized organs, I think we'll still need to do some more preclinical work.
Okay, thank you very much, Dr. Güell. That's all the time we have for today. I want to thank attendees and thank you, Marc, for the presentation. This concludes our session.